Very good morning, students. Welcome to biology classes. Today we are going to see 11th science biology and we are going to resume the chapter 14, that is respiration in plants. Right? We have already covered so many topics of this chapter, and yet we are going to see the next topic, and that is terminal oxidation. Right? So terminal oxidation is the name of oxidation found in aerobic respiration that occurs towards end of catabolic process and involves the passage of both electrons and protons of reduced coenzyme oxygen to produce water. Right? Now terminal oxidation, right? The word itself says the name of oxidation, right? See, this is oxidation which is found in the aerobic respiration, that is, which is occurring in the presence of the oxygen, right? And this is going to occur towards the end of the catabolic process, right? That is, here the whatever catabolic processes are taking place, that is all occurring, or every catabolic process is being turning, or this terminal oxidation is occurring towards the end of the catabolic processes, right? Also, it involves the passage of two things. The first is the electrons and the second is the protons, right? And these two of them are responsible for carrying out or for the occurrence of the whole ETS, and that is electron transfer system. Right now, in this terminal oxidation, we are going to see. Now, you have seen that when we saw about the citric acid cycle, whatever NADH2 were reduced, right? Those NADH2 is going to be entered inside the mitochondria. Now that was occurring inside the cytoplasm, but when it is entering inside the mitochondria, in mitochondria there are, now you know that mitochondria is double bound or double membrane organelle, right? So the difference, so here as we know that the mitochondria is a double membrane organelle, right? So there will be presence of, see here you can see the inner, so this is the inner mitochondrial membrane, right? So the membrane which is present inside, right? And there is presence of a outer membrane also. So in between the inner mitochondrial membrane and the outer mitochondrial membrane, there will be presence of a space which is known as intermembrane space. So this intermembrane space you can see over here. So at here there would be presence of the outer mitochondrial membrane, then the inner membrane space, the intermembrane space, then there will be presence of the inner mitochondrial membrane, and inside the mitochondria there will be presence of the matrix region. Now whatever NADH2 which were released inside the citric acid cycle, or we can simply say as TCA cycle, those NADH2, right, they are coming or they are entering inside the mitochondrial matrix, right? In matrix you are having NADH2. This NADH2 is going to be oxidized, right? So the oxidation is going to be take place, and here there will be an enzyme which is known as NADH dehydrogenase. D means removal and hydrogenase means the removal of the hydrogen molecules. So here from this NADH2 there will be the removal of the two hydrogen molecules, and those are the nothing but the protons. So this NADH is going to be oxidized, and the removal of the H plus occurs, and for this reason this NADH2 gets converted to NAD plus. Now when this NADH2, the oxidation, when the oxidation is taking place, what happens? It releases the two protons, and along with those two protons, right, there it is also going to release two electrons, right? So along with the protons, the electrons are also removed or released, right? So here this is the first complex, which is, you can see over here, which is the FMN. This, see here, this is inner mitochondrial membrane, right? So this all are nothing but the proteins which are present. They are simply said to be the carriers, right? Carriers means they are responsible for carrying this particular protons and electrons, right? Now the function of this, all the proteins which are present inside the membrane. Now, as they are present inside the membrane, they are said to be the integral proteins, right? So, their function of this integral proteins is to carry the protons as well as the electrons and to pass from one carrier to the next carrier, right? Now the protons which were passed from here, it is passing or it is being transferred from the matrix to the intermembrane space that you can see over here, right, where I'm showing the cursor. This has been passed with the help of the FMN. FMN is also one of the carrier which is responsible for carrying the two protons, and it is bringing those two protons or two hydrogen molecules in the intermembrane space. Now, after the release of the two hydrogen molecules or the protons, right, with the help of the NADH dehydrogenase, right, two electrons are being released out, right? So those two electrons are being accepted or they are passed to the FES, right? And further it moves to the UQ, right? UQ is ubiquinone, right? Ubiquinone is what here from this is the second complex which is observed here. So in the second complex, again two hydrogen molecules or two protons are being passed. So here how it is passing? So FES is responsible. They are the carrier for the electrons, right? They are removing or they are carrying the electrons, and the function of this FES is to carry the two electrons from FMN, and it is going to take those two electrons and bring up to the ubiquinone, right? So now the ubiquinone is having the two electrons, but side by side from the matrix the two another H plus ions, or we can simply say the protons. So two protons are going to be accepted by the ubiquinone, and this ubiquinone is going to be form the UQH2. So this UQH2, that is ubiquinone, with the protons, they are going to pass this protons from the matrix to the intermembrane space. So now this is the second complex which is responsible for passing of the protons from the matrix to the intermembrane space. Now another those two leftover electrons, they are passed to the cytochrome B, which is simply denoted as CYB, right? So this cytochrome B, this is going to pass again these two electrons to the next two carrier, that is FES. And further again it is going to pass in the third complex which is said to be the UQ, and that is ubiquinone. Again in this part also from the matrix, again the two protons, see over here you can see is that the particular two hydrogen molecules, right, over here see the arrow you can see is, right, this is going to enter, and for this reason this two molecules of hydrogen or the protons are being passed from the matrix to the intermembrane space, right? So here upon the passing of the two protons, the electrons are released. So from the UQH2, that is ubiquinone with the protons, after the release of the proton molecules inside the intermembrane space, two electrons are released and it is entering into the cytochrome C1. Further, it passes with the cytochrome C, and after that the cytochrome A to A3. And where here half molecule of water as well as two protons will be entered inside the middle, that is the inner membrane, and from the inner membrane there will be the release of water molecule, right? So here two things is occurring in the last, in this chain at the last, that half the water molecule, half oxygen molecule, as well as two proton molecules, they are being entering inside the inner mitochondrial membrane, while it is going to release one molecule of H2O. So this way the passing of the electrons takes place, and as the electrons are being passed, this chain is known as, or this system is known as ETS, that is electron transport system, right? And this is responsible or it is useful in the transport of the protons as well as the electrons. So here the oxidation which was taking place of the NADH2, to NAD, this whole sequence or the whole chain is running, right? So the electron transport chain says that it is a metabolic pathway through which the electron passes from one carrier to another inside the inner mitochondrial membrane, which is called ETC or mitochondrial respiratory chain. So this is a type of a metabolic pathway, and through this pathway the electrons are being passed from one carrier to the another, and this takes place inside the intermitochondrial membrane. As we have seen in the figure, this is the inner mitochondrial membrane in which the electrons are being transferred or they are being passed from one carrier to the next, and for this reason this is known as electron transport chain or electron transport system, or simply it is also known as mitochondrial respiratory chain. Now, the electrons from NADH produced during the citric acid cycle are oxidized by NADH dehydrogenase, and electrons are transferred to ubiquinone located within the inner membrane. Ubiquinone also receives electrons from FADH2, which is transferred to cytochrome C via cytochrome BC1 complex. As we have already seen over here also, this particular 2H plus ion. See, you can see over here, there were entry of the protons from here also and here also, right? So one is coming from the NADH2 and the second one is coming from the FADH2. So ubiquinone is the uh, carrier, right, which is going to be present inside the inner membrane of the mitochondria. It receives the electrons from FADH2 also, and these electrons are being passed or transferred to the cytochrome C with the help of the cytochrome BC1 complex. Now, when the electrons pass from one carrier to another via electron transport chain, they produce ATP from ADP and inorganic phosphate. Now, when the carrier are passing or transferring the electrons from one carrier to the next carrier, at that moment the ATP energy is produced, right? So from ADP, that is adenosine diphosphate, the ATP is formed, which is known as adenosine triphosphate. As well as along with the ATP, inorganic phosphate is also ADP plus the inorganic phosphate, they both combine to form one ATP. So the number of ATP molecules which are synthesized, it depends upon the electron donor. So how many electrons are being donated? This electron transport chain is based upon the number of ATP molecules which are being produced based upon or depends upon the electron donor. If more amount of electrons would be there, then more amount of ATP molecules will be produced. But if less number of electrons are there, then less amount of ATP molecules are produced. Now, the oxidation of one molecule of NADH gives rise to three molecules of ATP. So you can see over here that in from the citric acid cycle, when one molecule of NADH gives rise to three molecules of ATP, while oxidation of one molecule of FAD2 produce two molecules of ATP. So NAD give rise to three molecules of ATP, while FADH give rise to two molecules of ATP. This can be asked in MCQ, that when oxidation of one molecule of NADH occurs, how many molecules of ATP are produced? So when NADH is oxidized, then three molecules of ATP is produced, and when the oxidation of FADH2 takes place, at that time two molecules of ATP is produced. Now we are going to see about the oxidative phosphorylation and the phosphorylation, right? So we are going to see the difference between the oxidative phosphorylation as well as the phosphorylation, right? So oxidative phosphorylation means this occurs in the respiration process, right? So oxidative phosphorylation takes place only when the respiration process is taking place. While the photophosphorylation, right? Photo means light. So when the photo, that is light, when light comes, the first thing that clicks in your mind is what? The photosynthesis. So oxidative phosphorylation is occurring when the respiration is taking place, while the photophosphorylation is occurring when the photosynthesis is taking place. The second difference is the oxidative phosphorylation says that the energy of oxidation and reduction is used for production of proton gradient required for phosphorylation, right? So for getting the phosphorylation, right, for production of the proton gradient, that is balancing of the H plus ions, right, the energy of the oxidation reduction, that is the oxidation and reduction reactions are taking place in order to produce or in order to get a proton gradient which is required or which is necessary for carrying out the phosphorylation process, while the light energy in the photophosphorylation is utilized for the production of proton gradient for phosphorylation. So over here energy is being used, okay, and the oxidation and reduction reaction is taking place. But while when we are talking about photophosphorylation, the light energy is being used for the production of the proton gradient, right? So this is the difference between the oxidative phosphorylation and the photophosphorylation, right? So next is the, you can see over here is a figure, right? This figure says that the energy released during the ETC is used to make ATP with the help of ATP synthesis. Now, whatever electron transport chain, right? Whatever ATPs are produced, right? Those ATPs are being produced with the help of a enzyme or with the help of ATP synthesis. And this ATP synthesis consists of two major parts. The first is the F1 part, which is where I'm showing the cursor, the round colored shape, which is you can see over here, that is the F1 part. And the second, which you can see in the membrane, this is the F0 part, right? So there are two major parts of the ATP synthesis. The yellow colored portion is the ATP synthesis. Amongst them, this inner, inside the membrane, whatever is present, whatever part is present, that is F0 part, and at outside in the matrix whatever a round colored shape is there, that is F1 part, right? Now the F1 is a peripheral membrane protein. Now F1, as it is not present inside the membrane, as it is present insidely, right, attached with the membrane, for this reason this type of membrane is known as a peripheral membrane protein complex, and this is going to have a site for synthesis of ATP. So you can see over here, this is the site where the ADP and along with one phosphate, right, these two of them combine with each other along with the ATP synthase, it is going to produce ATP, right? So this is the site of the ATP synthesis from the ADP as well as the inorganic phosphate. Now, F0 is the integral membrane protein. Now, integral membrane protein, that means this type of protein is present inside the membrane. As you can see, this is the outer side. So, definitely the blue colored part is what the inner mitochondrial membrane, right? So this inner mitochondrial membrane consists of the F0 part of the ATP synthase. So this is said to be the integral membrane protein, and it acts as a channel for the proton. So this is responsible for passing the proton molecules, right? And this for each ATP produced, right, 2H plus passes through F0 from the intermembrane space to the matrix down the electrochemical proton gradient. So whatever intermembrane space is present in that, proton molecules are there. So from the proton molecules, when the two, see, if you want to prepare or if you want to synthesize one ATP molecule, from here two hydrogen molecules should be passed from the intermembrane space to the matrix region. So this 2H plus, or we can say the protons, this 2H plus passes from the F0 part and it enters inside the F1 part, where this ADP and the inorganic phosphate, these two of them combine with each other, and in the presence of these two proton, it produces the ATP energy, and this way the proton gradient is balanced. So this is all about the electron transport chain, right? I hope you are very clear with today's topic, and I hope to see you again. Thank you.